Welcome back to Pharma Show by Seiji. Today we're talking about one of the most important concepts in sterile filtration, the bubble point test, also known as BPT. Let's understand it in the simplest and clearest way. What is the bubble point test? The bubble point test is a non-destructive filter integrity test performed to ensure that sterilizing grade filters, usually 0.22 m or 0.2 m membrane filters, are intact and functioning properly. In simple words, it checks whether the filter pores are not damaged, whether the filter can truly remove bacteria, whether the sterile filtration step is valid. It is performed before and after filtration, especially in sterile drug manufacturing. Regulatory bodies like FDA, EMA, and USP consider it an essential filter integrity test. Why is bubble point test important? Filtration is a critical control point in aseptic processing. If the filter is damaged, microorganisms can pass into the final product, which is unacceptable. Bubble point test ensures the filter is not torn, broken, or defective, the pore size is consistent, sterile filtration is validated, batch release is safe. A failed bubble point means the entire batch may be rejected. Principle of bubble point test. Every filter membrane has tiny pores filled with wetting liquid, usually water. To push air through these pores, you need a certain minimum air pressure. This minimum pressure at which the first continuous stream of bubbles is seen from the membrane surface is known as the bubble point. The bubble point depends on pore size, surface tension of the wetting liquid, contact angle, smaller pores higher bubble point pressure, larger pores lower bubble point pressure. So, by comparing the measured bubble point with the manufacturer's specifications, we know whether the filter is intact. How the bubble point test is performed, step by step. Step 1. Wet the membrane. The filter cartridge or capsule is fully wetted with purified water or buffer. Step 2. Apply air pressure. A regulated pressure source, like compressed air or nitrogen, is used. Step 3. Increase pressure gradually. The pressure is increased slowly while monitoring bubble formation. Step 4. Observe the first continuous stream of bubbles. This is done through heavy checkmark automated integrity tester, heavy checkmark visual observation. Older method. Step 5. Record measured bubble point. Compare the value to the minimum acceptable bubble point provided by the filter manufacturer. Acceptance criteria. Each filter has a specification. For example, 0.22 m PVDF/PS membrane, 50-55 psi, 0.2 m nylon membrane, 42 to 48 psi. If the measured bubble point greater than or equal to minimum specification, the filter is pass. If it is lower, the filter is fail, indicating membrane damage, improper wetting, defective filter. Applications in pharma. Bubble point test is performed for sterile filtration of injectables, biopharmaceutical filtration, proteins, vaccines, aseptic filling line, buffer and media filtration, final product container closure. Validation. It ensures sterility assurance and is mandatory for every batch. Common problems: troubleshooting, low bubble point, fail, possible causes, filter damaged, improper wetting. Wrong installation, leaks in filter housing, instrument calibration issue, high bubble point, possible causes, dry spots on membrane, excess wetting liquid, wrong pressure ramp speed, no bubbles observed, pressure supply blocked, tester malfunction. Documentation. Every BPT must have batch number, filter serial number, pre-post-use bubble point, accept/reject, operator reviewer signatures, equipment ID. This becomes part of batch release documentation. Conclusion. The bubble point test is a simple yet powerful tool that ensures the safety of sterile drug products. A correct and validated BPT guarantees that filtration worked properly and contamination risk is minimized. Don't forget to like, share, and subscribe for more pharma knowledge.